Very good morning to all the EAPCET aspirants. This is your Prasanna ma'am from NEET World, and here we are to solve the EAPCET previous year questions on cell cycle and cell division, after which we will be starting the chapter of biomolecules. So yes, without much delay, let's get started with cell cycle and cell division EAPCET previous year questions. So if you look at the first question, where it is very clear and it is about what type, assertion and reason type of question. If you clearly look into assertion, where in assertion it is clearly said that syncytium is the result of karyokinesis not followed by cytokinesis, which is 100% correct. Okay? Wherein if you see, syncytium is the result of karyokinesis which is not followed by cytokinesis. In reason, in plant cells, middle lamella formation starts from the center, grows outwards of the cell, leading to the cell wall formation. That is what we have learned, right? Middle lamella, which is the first formed layer. At the center, this will move from the center to the periphery, forming up what? Cell plate, and middle lamella, which is the first formed layer of the plant cell wall. So here assertion is true and reason is true, but nowhere reason is explaining the assertion. In this case, what answer we have to choose? Option B. Look at the question properly. If you are not getting anywhere, you can type a message in the chat box. I will be addressing that. Fine, right? Yeah. Next, if we see second question, question 2. Cell that does not divide further due to the following changes in the cell cycle. So, whichever cells which are not often dividing, for example, if you can take nerve cells or cardiac cells, these are not often dividing, right? So, these cells, if they want to stop the cell cycle, they will stop at G1 phase. Okay, they will stop at G1 phase. From G1 phase, they will move to G0 phase. Fine, right? From G1 phase, they will move to G0 phase, and again when they are resuming back to their cell division, they will come back to G1 phase again. So, G0 phase is addressed as what, ma? Quiescent center, where cells are metabolically active over here. So, here if you see the cells that does not divide further due to the following changes. They will exit which phase? They will exit G1 phase and they will enter into S phase. Wrong. They will exit G1 phase and they will enter into G0 phase of the cell cycle. Option C is the right answer over here, which is statement based question. Okay. And next, if you have to observe question 3. Okay. Identify the meiotic stage in which homologous chromosomes separate while the sister chromatids remain associated at their centromeres. Okay, here sister chromatids will remain associated at the centromeres, right? So, this type of movement is what? Without centromere splitting. Identify the meiotic stage in which homologous chromosomes will separate. Here homologous chromosomes will separate without splitting of centromere, right? Wherein sister chromatids remain associated. So, this moves towards this pole and this moves towards this pole without any centromere splitting. So, there is no centromere splitting. This happens in which phase? Anaphase 1. Okay. Option C, this happens in anaphase 1. Identify the meiotic stage in which homologous chromosomes will separate. These are homologous chromosomes. They will separate. Wherein sister chromatids, they will remain associated at their centromeres. Fine, right? Each sister chromatid will move towards the opposite poles. There is no centromere splitting. Here the right answer is option C. Whereas in anaphase 2 or anaphase of mitosis, centromere splitting occurs, wherein sister chromatids will separate. Okay. Sister chromatids doesn't separate only in the anaphase 1. Clear, right? Clear till here? Any doubt anywhere, ma? Yeah. Here we go with match the following type of question. Observe clearly the events and the phases. Crossing over or recombination will occur at which phase? Crossing over or recombination will occur at Pakitian stage. So, A will be 4, right? Synapses, formation of synaptonemal complex, homologous chromosomes, bivalent or occurrence of tetrad. This is observed in zygotine, that is option 3. Fine, this is observed in zygotine. Next, weakening of synaptonemal complex. Dissolution of synaptonemal complex is the beginning of diplotin stage. Okay, so this will be option 2. Dissolution or disintegration of synaptonemal complex occurs in diplotin stage. This is the beginning of diplotin stage. Next, if you see terminalization of chiasmata, here he has just mentioned terminalization. This occurs in diakinesis, which is the last substage of prophase 1. Okay, this is the last substage of prophase 1. If you clearly look into the options in the proper order, first crossing over or recombination in pachytene, so A is for synapses, in zygotine option 3, weakening of synaptonemal complex is the beginning of diplotin, which is option 2. Next, D, terminalization, that is diakinesis, which is the last stage of prophase 1. So till here it is clear right now. Next question, if you have to observe, yeah, here again match the following type of question. So here if you look into the pattern of questionnaire that are being appeared from cell cycle and cell division, the questionnaire is mostly statement based, match the following type of questions. So here if you see separation of homologous chromosomes, separation of homologous chromosomes, this happens in which phase? Anaphase 1, wherein sister chromatids will move towards the opposite poles. Okay, this occurs in anaphase 1, which is option 3. Fine. Initiation of chromosomal pairing. Chromosomal pairing occurs in chromosomal pairing is nothing but formation of homologous chromosome, that is nothing but synaptonemal complex, which is synapses option 4. Fine, right? Chromosomal clustering at opposite spindle fibers. Chromosomal clustering means they are again decondensed to the chromatin back. So they are again condensed to chromatin, which occurs in the last stage of karyokinesis, that is telophase, which is option 1. And then disc shaped part of centromere. Disc shaped part on the either sides of centromere is nothing but kinetochores, which is option 2. Fine, right? So A is 3, B will be 4, C is 1 and D will be 2. So the right answer is option B. Clear till here? Any doubt anywhere, ma? Yeah. Next we will move on to this question, which is again assertion reason based question. So here if you see in assertion, the amount of DNA in cell undergoing mitotic cell division is doubled during the S phase. Yes, right, amount of DNA is doubled during S phase. So assertion is true over here. And then if you observe reason, in reason it happens due to doubling of the chromosome number in the S phase of interphase. Wrong, because in S phase what happens? In S phase DNA doubles, whereas number of chromosomes remains same. Fine, right? DNA doubles but number of chromosomes remains same. So here assertion is true but reason is false, where option C is the right answer. Clear till here, ma? Yeah. Next you look into this one. If onion root tip cell has 14 chromosomes and DNA content is 2C, okay, if it has 14 chromosomes and DNA content is 2C, what is the number of chromosomes and DNA content during G1 S phase and G2 phases respectively? Okay, so the number of chromosomes in G1 phase is how much, ma? 14 chromosomes and 2C DNA, right? So in S phase what is happening? Chromosome number remains same but DNA gets doubled. DNA gets doubled to what? 4C. Here in G2 phase again, number of chromosomes remain same, but DNA is doubled in the S phase. It will be same in G2 phase as well. So here it is 14 chromosomes 2C, 14 chromosomes remain same all across G1 phase, S phase and G2 phase. DNA amount doubles in the S phase, it remains same in G2 phase. So here the right answer is option A. Clear, right ma? This type of questions can be asked even by using graph. Okay, it can be asked by graphical representation as well. So remember the keynote or the point, which is a hypothetical statement only, because chromosome structure, it is evident in the metaphase. Clear structure of the chromosome, you can see it in metaphase. But then to make you clear that only DNA amount is doubling in the S phase, this hypothetical statement is used, that chromosomal number remains same in the S phase, but the amount of DNA doubles. Okay. Here if you see the next question, during metaphase, spindle fibers attach to what? Kinetochores. Spindle fibers attach to kinetochores. Where centromeres split and chromatids separate during which phase? Anaphase. Chromatids will separate during which phase ma? Anaphase. Clear, right? So during metaphase, spindle fibers will attach to kinetochores, centromeres split and chromatids will separate during anaphase. So here it is option C. Clear, right? Next question you look into, yeah, here statement based question again. Choose the incorrect statements. Whenever statement based questions are there, you have to analyze clearly whether they are asking you to identify incorrect statements or correct statements, and then you introspect the statements. First look at the questions properly and then you introspect the statements. Here if you see, cells of the lining of the gut are replaced by mitotic division constantly. True, right? Somatic cells or cell repair will happen because of mitotic division or equational division. Next, formation of liquid endosperm takes place if karyokinesis is not followed by cytokinesis. True, right? Here karyokinesis is not followed by cytokinesis, and then you can see the liquid endosperm. Here if you observe again the next question, in telophase chromosomes reach poles and they will start condensation or condensing to form chromatin network again. This is true, but here condensation is not happening. Chromosomes will decondense. Chromosomes will decondense to form chromatin network. In quiescent stage, the cell is metabolically inactive, which is wrong, because in quiescent stage, the cell is metabolically active. Cell is metabolically active. Fine, right? So here what are the incorrect statements, ma? Here this gets decondensed to chromatin, not condensation. So C and D are incorrect, which is option C. Clear, right? Next, choose the correct statements. We need to choose correct statements. Stage between two meiotic divisions. Okay, what is the stage between two meiotic divisions? Just look into this question, ma. Fine, right. Did you look into the question properly? Yeah, here we go. Stage between two meiotic divisions is called as interphase. True, right? Interphase is in between two M phases. During zygote, pairing of homologous chromosomes and synapses takes place. True. Synapses and pairing of homologous chromosomes takes place in zygote. Next, initiation of mitotic spindle occurs during metaphase. True, right? Initiation of mitotic spindle will occur during metaphase. Correct. In animals, mitosis occurs in diploid somatic cells. Fine, right? In animals, mitosis will occur in diploid somatic cells and not in haploid cells. Fine. So, here we need to choose the correct statements. If you observe clearly, stage between two meiotic divisions is interphase. A is true. And then during zygote, pairing of homologous chromosomes takes place. True. Initiation of mitotic spindle occurs during metaphase. It occurs during late prophase only. Okay. And in animals, mitosis occurs both in the haploid and diploid cells. Okay, not just in the diploid somatic cells. So, the correct statements goes option A and B. Okay. Fine, right? Next, just look into it once again. The stage between two meiotic divisions, that is in between Meiosis I and Meiosis II. This stage is addressed as interkinesis. This is not interphase, okay. And BUC, during zygote, pairing of homologous chromosomes takes place, that is synaptonemal complex, formation of synaptonemal complex. Initiation of mitotic spindle occurs during metaphase, yes, during late prophase or the beginning phase of the metaphase, and mitosis occurs both in the haploid and diploid cells. Okay, so here the right answer will be B and C. Okay, sorry for the slight inconvenience. Here the first, it will be interkinesis, not interphase, and mitosis in animal cells occurs both in the diploid and haploid cells. Clear, right? Yeah, yeah. Next question. This one, if you see, question wherein this is numerical based and this is based on chromosome number. That is the reason you should be aware of the table which is given in reproduction in organisms. In reproduction in organisms, you have a table wherein you have a set of organisms, both plant and animal species, their haploid set of chromosomes and diploid set of chromosomes. This question is in alignment with the chromosome number. You see properly, chromosome number of potato gametes. Potato gametes means these are haploid. Diploid set of potato ploidy will be 46. Haploid set will be 24 chromosomes. Okay. This will be equal to double the housefly meiocytes. What is a meiocyte? The cell which is ready to undergo meiosis. Means it is double this housefly diploid cells. Okay. Diploid housefly is having 12 chromosomes. Double the diploid set is 2 into 12. That is equal to 24. And then if you see trice of fruit fly meiocytes. Diploid set of fruit fly is having 8 chromosomes. Diploid set of fruit fly is having how much? 8 chromosomes. Thrice of fruit fly, that is 3 into 8 will be 24. So, now we are clear with this. Haploid set of potato gametes, that is 24, is equal to double the housefly meiocytes, which is 24 again, and it is thrice the fruit fly meiocytes, which is 24. Clear, right, ma? Here you see. Find the haploid ratio of chromosome numbers. Haploid ratio of chromosome numbers. If you see, fruit fly haploid ratio, diploid is 8, haploid will be 4. Housefly, diploid is 12, haploid will be 6. Potato, diploid is 46, haploid is 24. Okay, 4 is to 6 is to 24, which is option C. Okay, option C is the right answer. Clear till here? Any doubt anywhere, ma? Yeah. Now we see here. Next question. Sequence of subphases of prophase 1. What are the substages of prophase 1? Prophase 1 will have 5 substages. First will be liptotene, followed by zygotene, then pachytene, then diplotene and diakinesis. You just observe in every consecutive paper, if you can see the ratio of questionnaire from the entire chapter of cell cycle and cell division, you can clearly notice that maximum amount of questionnaire has come from prophase 1 only. Maximum amount of questionnaire has come from different phases of prophase 1 and on the chromosome number which happens in S phase where the chromosome number is constant but DNA doubles. And one more thing, chromosome number and their ratios in between different organisms, both haploid set and diploid set should be in your mind. So here if you see the sequence of this properly, first is 3 followed by 1 followed by 2 followed by 4 and then 2 and then 5. Option C is the right answer. Clear everyone? Any doubts anywhere ma? Yeah. Next if we observe this chapter. So the complete questionnaire practice of cell cycle and cell division is done. Now we will move on to the major chapter, that is biomolecules, which is a hotspot and most important chapter, not just for your EAPCET but also for your NEET and for your IPE as well, where SAQs and VSAQs will be targeted from here. So that important chapter this is. Look into the fundamental insights of this chapter. What are the contents that you have to cover? First is elemental analysis and chemical composition. Okay, elemental analysis and chemical composition. Before entering into the fundamental insights, first I would like to know how many of you know what is meant by biomolecules. Okay, biomolecules are nothing but those constituents or organic molecules which are present in a living cell are addressed as biomolecules. Remember properly, those molecules or living components or cellular constituents which are present inside a living cell is addressed as what? Biomolecules. Here we go with the fundamental insights. First is elemental analysis followed by chemical composition of different molecules. Then you will clearly learn about the characterization and differentiation between primary and secondary metabolites, and then you will learn about biomacromolecules followed by what are included under biomacromolecules: proteins, carbohydrates, nucleic acids and lipids. Lipids, of course, they are not strictly biomacromolecule. I will tell you why and how. Next it is followed by enzymes. Enzymes, nomenclature and their mechanism of action. What will be the factors that are affecting the enzyme activity, its classification, nomenclature of enzymes. Lastly, cofactors. These are the fundamental insights of biomolecules, wherein there will be a definite question on the biomacromolecules or on the enzymes. Okay, that important it is. And if you consider nucleic acids, lipids, these components and all, nucleic acids, the questions will be connected with molecular basis and asked. These are the fundamental insights. You can take a screenshot of this and have a note of this. These are the fundamental insights. Cover this chapter point by point, segment by segment. Find right. Next, here if you see the elemental analysis of earth crust and in human body. Okay, elemental analysis elemental analysis in earth crust and in human body. If you clearly analyze and introspect, all the elements which are present in the sample of earth crust are also present in the sample of living tissue. But relative abundance of carbon, hydrogen, with respect to other elements is higher in the living organism when compared to the earth crust. This is a very important statement and liner from your text and from the NCERT. Okay? The relative abundance of what? Carbon, hydrogen with respect to the other elements is higher in the living organism when compared to the earth crust. As such, in the elemental analysis, a table is given out which is very important, where you have to clearly analyze. You need not to remember the exact number beta. Exact number you need not to remember, but then you have to know the ratio, like which element is maximum present in the human body and minimal in the earth crust as such. So, here if you see hydrogen which is 0.14 in the earth crust and 0.5 in the human body, which is more in the human body, and carbon also if you see its relative abundance is more in the human body, oxygen whose relative abundance is more in the human body, nitrogen relative abundance is more in the human body, sulfur the same, fine, right. Sodium if you see the relative abundance is more in the earth crust, calcium relative abundance is more in the earth crust, phosphorus it is high in the living tissue, magnesium more in the earth crust and silicon more in the earth crust. It is toxic and it will be negligible in the living tissue. This relative abundance you have to be clear on this note. Exact number is not required. Okay. Approximately you should have an idea like which element is relatively abundant in the human tissue, living tissue, or in the earth crust. Take a screenshot of this and refer this when you are going through the NCERT. Have the element, now you have got an idea on the elemental analysis. How the chemical analysis is performed, how you will get to know about the elements, chemical constituents, cellular constituents which are present in a living tissue. Okay, what is the procedure for that? How do you analyze a chemical composition? First you have to take a living tissue which is taken in a mortar by using mortar and pestle and by the chemical trichloroacetic acid. This is grinded. Okay, this is grinded by trichloroacetic acid. Okay, using mortar and pestle. After which you will get a thick slurry which is filtered using cheesecloth. After filtering it using cheesecloth, you will get two fractions of the straining. One is addressed as acid soluble pool and other is addressed as acid insoluble pool. After grinding it with mortar and pestle and filtering it with a cheesecloth, and when it is taken into a test tube, okay, two fractions are analyzed. One is dense and occupied at the bottom. This is known as retentate or acid insoluble pool, whereas the other which is over the periphery, okay, in the form of liquid, this is filtrate or acid soluble pool. This is the retentate acid insoluble pool, this is filtrate acid soluble pool. And next if you see, what are the components that you have identified in acid insoluble pool and what you have identified in acid soluble pool? That will be the question. Remember that in acid insoluble pool, fine, right. So, which is retentate. This is what? Acid insoluble pool or retentate. In retentate, if you observe carefully, all biomacromolecules are obtained. All biomacromolecules like polysaccharides, nucleic acids and proteins whose molecular weight is above 10,000 Daltons. Fine, the molecular weight of this molecules is about 10,000 Daltons. But then lipids whose molecular weight will not exceed more than 800 Daltons, these are present, these are obtained in the acid insoluble fraction. It is because whenever the cells are grinded by mortar and pestle, the cell membrane gets disintegrated, yes or no? When the cell membrane gets disintegrated, the lipid components present in the cell membrane will turn up forming liposome or into micelles. Okay, they will turn up forming what? Liposomes and micelles, and they will form into globules which are water insoluble vesicles. This turn up forming what? Water insoluble vesicles. fine, they will form water insoluble vesicles and they are obtained in the retentate, fine, right. And periphery, if you see, this is acid soluble pool or filtrate, acid soluble pool or filtrate, wherein the molecular weight of this will be, molecular weight of the chemical constituents present over here will be 18 to 800 Daltons. Okay, they will be from 18 to 800 Daltons, which are cytoplasmic composition. All the cellular constituents will land up here, okay, in acid soluble pool. So you are clear with this note, no? Retentate, acid insoluble, whose molecular weight, the components biomacromolecules here, their molecular weight will be about 10,000 Daltons, whereas lipids whose molecular weight is less than 800 Daltons, but then they will land up in the retentate because of the disintegration of cell membrane will turn up forming lipids into liposomes or micelles, okay, which are water insoluble vesicles. They will be landed up into retentate, and above which we have filtrate or acid soluble pool, molecules or cellular constituents which are present in the acid soluble pool will be from 18 to 800 Daltons. Clear till here? Any doubt anywhere, ma? Yeah. Next we will move on to identifying the dry weight. How you will get the dry weight of the components? You have analyzed elemental analysis, chemical analysis is done. Now ash analysis. Okay. Ash analysis. How ash analysis is performed? Ash analysis is performed like a small amount of living tissue, either it is a plant or animal weight living tissue. This tissue sample is dried and weighed again. Okay, this is dried and weighed again. When it is dried, it is fully burnt, wherein gaseous forms like carbon dioxide, water vapor are oxidized totally, and that ash whichever is present, that will contain inorganic elements like calcium, magnesium and silicon, etc. Those ash analysis will give you some elements or the components whichever are present in the ash. This is what ash analysis. Any dry material is taken, burnt completely, okay, where carbon dioxide and water will be evaporated totally, and the components whichever are remained in the ash are looked out. For example, sodium is one of the component, potassium, calcium, magnesium, water and other compounds include sodium chloride, calcium carbonate, phosphate and sulfates. Clear till here? Any doubt anywhere? Yeah. So all the carbon compounds that we get from the living tissue can be biomolecules. All the carbon compounds that we get from any living tissue can be what? Biomolecules. Ash will contain inorganic elements. Okay, ash contains inorganic elements like what? Calcium and magnesium. Okay, ash will contain this. Inorganic compounds like sulfate, phosphate, etc. are also seen in the acid soluble fraction. These are also seen in what? Acid soluble fraction. Where do you find inorganic elements? Cellular constituents. You will find them in acid soluble pool. Remember that carefully. In acid insoluble pool or retentate, you will get only biomacromolecules like polysaccharides, proteins, nucleic acids and lipids. Out of these four, the molecular weight, if you see, it will be 10,000 Daltons approximately for the other three, whereas 800 Daltons for lipids. Remember these points and statements carefully, ma. Okay. Now you see primary and secondary metabolites. Firstly, what is a metabolite? Metabolite is any component that is obtained in the intermediate pathways of any biosynthetic pathway or as the products of biosynthetic pathway. Okay, the constituents which are obtained in between the biosynthetic pathways as intermediates or at the end as products are addressed as metabolites. Metabolites, based on their function primarily, they are classified into primary metabolites and secondary metabolites. So, if you clearly see into the definition, metabolites are intermediates and end products of cellular metabolism. Okay, and organic compounds will include like amino acids, sugars, etc. are called as biomolecules, which are known as what? Metabolites. These are of two types, primary metabolites and secondary metabolites. Now we will look into the clear analysis in between primary and secondary metabolites. I hope the screen is clear to you, right? Yeah. Here we go with primary. Okay, those chemical compounds might be intermediary compounds or at the end products, whichever compounds are formed. If they are directly useful for growth and development of the organism, they are addressed as what? Primary metabolites. Okay, they are addressed as primary metabolites. Fine, right. Secondary metabolites, in analysis of plant, fungal and microbial cells, in analysis of these cells, several compounds are seen beside primary metabolites. Several are seen beside primary metabolites, and these are called as what? Secondary metabolites. Okay. Here primary metabolites, these are involved in primary metabolic process like respiration and photosynthesis. Secondary metabolites, they are not required for primary metabolic processes. Primary metabolites include carbohydrates, proteins, lipids, nucleic acids. Okay, these are the examples of primary metabolites. Secondary metabolites include alkaloids, flavonoids, rubber, essential oils and antibiotics and colored pigments as well. These are the examples of secondary metabolites. This is directly involved in growth. Okay, such compounds are highly useful for organism itself and are present from the starting of the reaction. If you see secondary metabolites, they have no direct role in growth and development. These compounds, they will have limited growth, limited role in the organism itself, and they are formed in a particular stage of the development only. Primary metabolites are in generous quantities and can easily be extracted. Secondary metabolites are in smaller quantities and it is difficult to extract them. It is expensive process. These are parts of basic molecular structure. These are not part of basic molecular structures of the cell. Are you clear with primary metabolite and secondary metabolites, ma? Next, secondary metabolites examples. This is given in your NCERT most important table. Most important table. So see it carefully. First, pigments. Pigments include many pigments like carotenoids, xanthophylls, anthocyanins. These are different pigments. Alkaloids include morphine, codeine. These are the alkaloids. Terpenoids include monoterpenes and diterpenes. Essential oils include lemongrass oil. Toxins, abrin and dricin. Lectins include concanvalin A. Drugs, vinblastine, vincristin and curcumin. Polymeric substances include rubber, gums and cellulose. So here if you observe clearly, there is a mnemonic which I have given where you will enjoy learning this. Pretty angry tigers eat tasty laddus during picnics. Of course, this sounds crazy, but you will remember it easily. P. Pigments, alkaloids, terpenoids, essential oils, toxins, lectins, drugs, polymeric substances. 2 to 3 examples for each of the secondary metabolite. Remember that. There is one feature common to all these compounds found in what? Acid soluble pool. There is one feature common for all these secondary metabolites, that secondary metabolites are identified in filtrate or acid soluble pool. Fine. Next, they have molecular weights ranging from 18 to 800 Daltons approximately. Okay. Next we go with this biomacromolecules. Macromolecules generally belong to how many classes? 4 classes of organic compounds we have discussed, right? First is polysaccharides, then nucleic acids, proteins and lipids. Chemical compounds without exception of lipids have molecular weights ranging from 10,000 Daltons and above in acid insoluble pool. Lipids are not strictly biomacromolecules. Okay, I already told you the reason, because their molecular weight is 800 Daltons. Clear till here, ma? And here if you see lipids, nucleic acids where we will come across nucleotides, carbohydrates, its classification, we will have monosaccharides, disaccharides, oligosaccharides, okay, and polysaccharides. Proteins where we will learn about primary structure, secondary structure, tertiary and quaternary structure of proteins. Okay, so all the biomacromolecules we will exercise in the next class. Today what we have dealt, ma, if we have to take a recap of this class, we have solved out all the EAPCET previous year questions of cell cycle and cell division, and then we have started with the biomolecules, fundamental insights. We have covered up elemental analysis, chemical analysis, followed by primary and secondary metabolites, and then we have a brief account on biomacromolecules as well. Fine, right? In the next class, we will have a clear note on biomacromolecules. Okay, so that's all for today's session, beta. Thank you.